All right. Well, I'd just like to start by thanking both Christoph and Axel for organizing this. I think it's been such a wonderful seminar series so far, and I just feel very privileged to be talking amongst all these great, great speakers. And yeah, so I'm going to be talking about some work going on in my lab. I'm currently at the University of Texas at Austin. I've been here since fall 2019. And this is my amazing lab here, my postdocs, Claire and Harry, and grad students, Leah and Smriti. Smriti was actually an undergrad in Axel's lab, so it's kind of nice that, yeah, that we can all come back together. Although I have to say, it being 7 a.m. here, I'm not expecting any of my lab to actually be here today. Okay, so I'm going to talk about 3 themes in my lab. The first being understanding how animals learn about multiple complex rewards, some experiments on comparative cognition in bees, and then finally anthropogenic effects on bees. Okay, so as Ellie mentioned last week, it wouldn't be a bee cognition talk if we didn't have a picture of von Frisch and his lederhosen. I'm going to give a very brief overview here because I'm sure you all know this, but of course we've been using bees to study cognition for a really long time. And von Frisch is kind of famous for his work demonstrating color vision via a learning experiment in honeybees. But somebody who received a lot less attention, who was recently highlighted in this nice paper by Danielle Lee in Animal Behavior, was Charles Henry Turner, who also did a series of experiments on color vision of the honeybee. And if you're somebody who likes kind of old school animal behavior experiments, I would highly recommend reading this paper. It's wonderful. Okay, so learning is critical for generalist bees like bumblebees. If you're, a bee that visits many different types of flowers, these flowers, they vary in how they look, how they smell, but also importantly in the rewards that they offer to the bees. And so it makes sense from the perspective of an individual that they'd be able to rapidly learn which of these flowers has got the best rewards. But it's also critical from the plant perspective. Plants have got all these scents and colors to attract bees to them, but also to make them more learnable to bees, because what's advantageous to a plant is that a bee will go to it, get some pollen on it, go to a conspecific and result in successful pollination. A plant doesn't want a bee wasting its pollen on a heterospecific. And we know that when bees like honeybees or bumblebees land on flowers, they can learn just about any feature to do with that flower. They can learn olfactory cues or visual cues like the color, the pattern, the shape of a flower, the temperature cues, texture cues, and even the electric field around the plant. But basically everything we know to do with learning in this foraging context comes from pairing conditioned stimuli like these ones here with nectar as a reward, as the unconditioned stimulus. And plants don't just have nectar, they have many different types of reward. They have resins or scents that bees collect. But the most prevalent reward, aside from nectar, is pollen. And so there are plants that bees only collect nectar from, but there are also flowers that bees only get pollen from, like this lupin or dodecathion. There are many flowers that bees collect both nectar and pollen from. And an individual bumblebee can collect nectar and pollen on a single foraging bout from a single flower. And so we tend to think of nectar mostly as carbohydrate and pollen mostly as protein, but these are complex rewards and they have a whole load of secondary compounds. I'm not going to really talk about this too much today, but we do have projects going on in the lab where we are interested in this more complex chemistry of these floral rewards. So pollen and nectar are really different types of reward for the bee. When a bee is collecting nectar, she ingests this sugary solution directly. Whereas when a bumblebee collects pollen, she's generally not eating it on the flower, she's scrabbling it into her corbiculae, into her pollen loads, and the pollen is taken back to the colony. It's popped off into the honeypots and it is fed to developing larvae. So these are two really different types of reward from the bee perspective, but they're also really different types of reward from the plant perspective. So nectar is kind of a relatively cheap reward for plants to produce. Its primary function is probably to function as a reward. Whereas pollen, it is used as a reward by many plants, but it's also, of course, the male gametophyte. And so what pollen is for a bee is different to what it is for a plant. And so there's this potential for conflict here, where what is advantageous for the bee is to get as much of this key nutritional resource as possible, take it back to the colony, feed it to your babies. But any pollen that is fed to larvae is essentially lost fitness to the plant. And so this conflict shows up in some pretty cool ways. Some pollen has got spikes on it, which makes it difficult for bees to pack it into their corbiculae. Other pollen is put into these little pollen sacs, these pollinia, and affixed to the back of bees, so the bees can't even access the pollen. And other pollen, or a lot of pollen, has got secondary chemistry, a whole load of chemical defenses. And a study that I'm not going to talk about today, but I thought I would just mention briefly here is we did one study that showed that bees actually taste pollen while they're collecting it, and the secondary chemistry can affect how much pollen they collect and then their subsequent choices when they forage. And so it does seem like plants might be using the secondary chemistry via taste in bees to limit pollen removal. Okay. And so overall, we actually know relatively little about how bees perceive or learn about pollen relative to our understanding of nectar. And so when we're doing experiments with bumblebees in the lab, which of course many of us do, they're kind of a model system now for understanding cognition, the question is how representative this is of how bumblebees actually forage when they're dealing with multiple rewards. And so I just wanted to give a shout out here to the work that came before my work, because people, who've already presented here and are involved with the symposium have done a whole load of work in both honeybees and bumblebees that really set the stage for the work that I'm going to talk about here. So the two experiments I'm going to talk about is, first of all, looking to see if bees could learn floral associations with the presence of pollen, and then how this might interact when they're learning about nectar as well. Okay, and so I've just talked about how great bees are at learning, so I just wanted to take a second and say why they might not learn about pollen. Because it might seem like this was a foregone conclusion, but actually when I started this experiment, I didn't actually know what we were going to find. So generally speaking, nectar is harder to find than pollen. It's often concealed inside flowers. Bees, when they land on a flower, don't often know what, from looking at it or smelling at the flower, how much nectar or the concentration of nectar that they're going to get. Whereas pollen, on the other hand, is often stuck, on the end of anthers where it's brightly colored or smells really strong. And so it could be that there are enough cues coming from the pollen directly that the bees don't have to learn associations with the other floral cues around it, right? It could just be that a bee approaches a flower and can see and smell whether there's pollen there or not. Okay, so in this experiment, we took Bombus impatiens, our kind of lab species, and attached a colony via a connecting tube to a foraging arena where we have one color of flower has got pollen on it, and the other color has got the scent of pollen, but no actual pollen. And we tag individual bees, and all the work is done using individuals. And so this is what this looks like. Here is a bumblebee collecting pollen off one of our fake flowers. This is cherry pollen, which we found the bumblebees really seem to like. So that's the pollen that I used in all these experiments. And so I'm going to show you a video now of what this looks like. A bee comes in and she can collect as much pollen as she likes from these yellow flowers that are rewarding. Like I said, the blue flowers, in this case, have got the scent, but no actual pollen. In this and in all the experiments I'm going to talk about, we always counterbalance for the color, of course. In this experiment, they had 2 training trials. And so each trial is essentially a foraging bout. The bee comes in, she can forage for as long as she likes. She leaves, goes back to the colony, takes off her pollen loads, returns for a second bout, goes back to the colony, and then comes back for a test trial. And in this experiment, and again, in all the experiments I'm going to talk about, these test or probe trials, the key thing is that there's no rewards on any of the flowers. So the anthers we used are scented, just like the unrewarding anthers here, but they can't be using like a visual cue of the pollen, for example, because there's no pollen in the test phase. Okay, and so here is a slowed down video of a bee during one of the learning trials. So you can see that she's searching on these blue flowers. She's not finding pollen there, and then she finds pollen on this yellow flower and stops to collect it. And now I'm going to show you a video of a bee during a test trial that was trained to yellow. And so just a reminder, there's no pollen on these flowers in the test trial. And so, yeah, you can see really clearly, right, that she's searching on those yellow flowers. And so this was really nice. It was really nice to see this behavior in such a kind of strong, compelling way. This is the data. So on the y-axis here, we have the first 10 landings that they make in that test phase. Bees that were trained to blue, mostly going to blue flowers. Bees that were trained to yellow, mostly going to yellow flowers. And you can see that this is a really strong effect, even with a pretty small sample size. In this setup, the color was meant to represent the flower's corolla. A lot of flowers, including boraginous species, like I've shown here, their corolla color can cue how rewarding their nectar or pollen is, like what stage the flower is at. But flowers also vary in their anther color. And so we wanted to repeat this, but this time varying the anther color. We found exactly the same thing. I don't normally mention this because I feel like it's kind of niche interest, but I thought this was really interesting that their color preferences are opposite for corolla versus anther. They love the blue corollas, but the yellow anthers, which kind of makes sense given that, anthers are normally yellow, but I thought that was kind of interesting. Flowers also vary in specific combinations of their corolla and anther. And so we also did a treatment where we had, these specific combinations and then tested bees with all 4 possible combinations. And so they didn't have to learn like both the anther and the corolla, but they did. They learned the specific combination, no problem. And then for another group of bees, I trained them and I tested them either after a day or after 7 days. And I found that they remembered this association both after that day and also after 7 days. And so I was pretty excited about these results because really it means that when a bee comes into a meadow like this, she's making her decisions of which flowers to visit based not only on her memory of where she found nectar in the past, but also on her memory of where she found pollen in the past. And potentially, for a long period of time, right? For like potentially a week or so, which for an animal that only lives for a few weeks to a couple months is actually a significant portion of its lifetime. Okay, so like I mentioned, many bees, including bumblebees, will collect both nectar and pollen at the same time. And so I also wanted to look at learning about these 2 rewards concurrently. And in learning theory, we tend to think about there being a stimulus and an unconditioned stimulus. But, and a lot of learning theory actually is based around a single reward with multiple conditioned stimuli. So for example, when we're thinking about questions to do with multimodal cues or the timing of different conditioned stimuli. But in this case, and in many cases in animal learning, there are actually multiple rewards for a given stimulus. And I have gone down, into the experimental psychology archives to really try and find other examples of experiments looking at this. And I haven't found many good ones. And so if anybody has any suggestions, I really would love to hear about it. But with bees, and like I said, with animals more generally, we're often having to learn about multiple rewards for a given stimulus. And so one thing that I'm trying to do now with the system is using this setup to look at that more generally. And so a bee, for example, in this case, might learn, okay, yellow flowers are rewarding. There's going to be a reward there. I go to this stimulus. But what if there are these different rewards that are in conflict with each other? So now this flower, for example, is rewarding for pollen, but it's not rewarding for nectar. And this blue flower is the opposite. The bee can't just learn that one stimulus is rewarding and one isn't. It has to learn a specific response for a given stimulus. And so I did this experiment using exactly this setup. This is what this looks like. So here is a bee collecting nectar from a yellow flower. So there's nectar in that nectar well. There's no pollen on that flower. And then she goes to the blue flower, she collects pollen, and she probes that nectar well, but it's unrewarding. She's just getting water there. So that's kind of an error that she just made. And so the nice thing about this setup, as you can see, is it's really easy to discriminate what the bee is expecting to find, right? What is she searching for in terms of these two different rewards? This time, I gave these 6 training trials, followed by a test trial where they both had the S+ and the S-, but they also had 2 novel colors, the purple and the orange here. And when we plot these colors into bee color space, taking into account their spectral sensitivities, basically this tells us that all these 4 colors are very discriminable to a bee. They can tell the difference between these colors, but the purple and the blue are relatively closer to each other, and the yellow and the orange are relatively closer to each other. And that will be relevant in a minute. Okay. So first of all, they had no trouble learning these 2 different associations. When the yellow flowers had nectar, they're searching more for nectar on the yellow flowers, and they're searching more on the blue flowers for pollen. With the treatment, they were trained to the opposite association. They're doing the opposite. So we can see really clearly, okay, they've learned to go to the correct color for the correct reward. But they also went a ton to these novel colors. They essentially treated purple flowers as if they were blue and treated orange flowers as if they were yellow. And so we know from a bunch of previous work that the bumblebees do generalize their responses a lot, unless you explicitly train them not to. But the thing that I thought was really cool here is that they're generalizing as much for pollen as they are for nectar, and they're also generalizing in this very kind of reward-specific way. And so the implications of that are basically, okay, imagine a bee, you're foraging in this meadow and you're going between the sun and the shade. The flowers are going to look a really different color in these 2 different light conditions. But if you're collecting pollen from a lupin in the shade, you go and approach your lupin in the sunshine, and you're still expecting to find pollen there. So there's implications in terms of thinking about foraging intra-specifically in terms of the plants. But also I think there are implications in terms of thinking about interspecific relationships with plants. So say our bumblebee approaches this tomato flower in your backyard and it's never seen a tomato flower before in its life. We know that these buzz-pollinated flowers often have features in common with each other. And so there's a good chance that a bee will approach this flower with the expectation of finding pollen there, even if she hasn't encountered this plant type before. Okay, and so I then wanted to know, okay, they can learn these 2 different associations at once, but is there a cost to learning? And in a treatment I did where bees they collected pollen from all the flowers and only 1 color had nectar, there was no effect on their ability to learn that nectar association. But the reverse was not true. When there was nectar on the flowers, it impaired their ability to learn about where to find pollen. And the thing that I thought was really interesting here was that it didn't matter which flower the nectar was on. So this is the test phase results, the average number of correct visits in the test phase. Bees that are just learning that one of the flower colors has got pollen, they're getting about 90% correct. When the same flower that had pollen had nectar on it, or when the other flower had nectar on it, there's no difference between these treatments. They're both worse. The bees are performing worse than the pollen-only treatment. It doesn't matter which flower has got the nectar on it. And so I don't think that this is an interference thing. I think that this is a motivational thing. And the reason I say that is because the bees that have the nectar on the flowers are also collecting about half as much pollen as when there's only pollen on the flowers. And so something about the presence of nectar is just making bees much less motivated to both collect pollen, but I also think to learn about where the pollen is. And if we go back to thinking about this bigger picture of what is pollen to a bee and to a plant, from the bee perspective, it seems like nectar is a much more salient reward to them, which is why it's having these effects on their learning. But from the plant perspective, it also makes sense that this cheaper reward would be more salient, because by rewarding bees with this kind of sugary crack treat, they're essentially distracting them from the more kind of valuable resource. So by having nectar there, the bees are removing less pollen than they would be otherwise. And I just wanted to mention briefly that Jake Francis, who is now a postdoc in UC Davis, did an experiment after this, also showing that when plants add chemical defenses to their pollen, and bees are less likely to want to collect it, this can also be offset by adding nectar as a sugary reward. And so this is kind of further evidence for plants using nectar as a way to manipulate bees' behavior when it comes to pollen. Okay, and so in 2020, I was awarded an NSF grant to follow up on some of these questions. And so, as I mentioned, we're using pollen and nectar and bumblebee foraging to more broadly understand how animals make decisions when there are multiple types of reward. And my grad student Muth and postdoc Claire are both working on these questions now. So a couple ones, for example, that we plan to work on or are currently working on are how animals compare options where you're dealing with multiple axes of quality. If you have a flower over here that's got great nectar and not so good pollen, how do you value that relative to a flower that's got great pollen and not so good nectar? Another thing we're looking at is how comparative decision-making is affected by multiple reward types. We know a lot about comparative decision-making when it comes to a single reward, but how animals integrate these different axes of reward quality. Okay, so now I'm going to talk a little bit about using bees to ask questions in comparative cognition. So in thinking about how environments shape cognition, there is a lot of classic work on this, right? So one classic example comes from our voles, where species that are polygamous, where the males have got larger home ranges, also have better spatial performance relative to the females compared to monogamous species that have similar size of home range and more similar spatial cognitive abilities. Another example that I really like is from Vladimir Pravosudov's group with caching chickadees, where the chickadees that cache in conditions where there's a greater requirement for them to remember where their caches are also have superior spatial abilities, and you see this difference also represented in the hippocampus. So we obviously know that environments shape cognition. I think a lot of the examples do come from spatial cognition because of the kind of dramatic differences we see represented in the animals' ecologies. But I also think that bumblebees And bees in general offer a really useful system for asking comparative questions. I'm going to focus on bumblebees here, but something that we're doing more broadly in my lab is trying to branch out into other bees, because there's just so many bees out there, right? 20,000 species worldwide, and that vary so much in their ecology and their behavior. But just sticking with bumblebees for now, we all know, of course, there are model species for studying cognition, but of course, most of the work that is done on two species, Terrestris and Impatiens, and on one particular stage of that colony life cycle, the forager stage. But there are so many species out there. These species vary in their ecology, both within and across species. And it's relatively easy. This is the key thing, I think, is that bumblebees are everywhere, and they're even the wild bees are just highly motivated to participate in cognitive tests. And so here is an example of how we do this. So this is looking at color learning in a bumblebee. So our bumblebee here, there's a reward on that blue strip of paper, there's sucrose. She went to that strip first, so that's a correct choice. We give her that for 3 seconds, and then she gets 3 seconds with the yellow strip, which is unrewarding. So we do these repeated trials. I'm going to show this is now one where she makes an incorrect choice. So she goes to that unrewarding strip before finding it on the rewarding strip. So this protocol is just a nice way where we can control the timing of stimulus presentation, right? In a free-flying assay, the bees are deciding when they want to land on flowers, whereas now we can control aspects like the intertrial interval and other things that we know are relevant to learning. But the bee is still free moving. And so there's, things that we can get at that we can't get at in a PER setup. Okay, so before I go into this experiment, I'm just gonna remind everybody just quickly of the bumblebee life cycle. So queens overwinter, they then emerge in the spring. And when the queens emerge, they're essentially functioning as solitary individuals. They're foraging for nectar, they're finding their location that they're going to start their nest. And once they start their nest, they're going to be foraging for pollen. But really, all the pressure at this stage is on this one individual not to mess up, right? Because if she messes up, if she dies, if she doesn't find a place to start her colony, then there's going to be zero fitness. Once she starts her colony, then she switches role into becoming basically solely reproductive. So once she's producing forages, then she stays in the colony, she becomes this egg-laying machine, and the foragers are the ones going out and collecting the nectar and the pollen. At the end of the colony life cycle, new queens or gynes are produced, males are produced, they leave the colony, they mate with other queens and males, then the males die, the gynes overwinter, and the whole thing starts again. And really, basically everything we know about cognition comes from this one stage, right? We tend to look at the foraging females at this one stage of the colony life cycle. One thing I got interested in a few years ago was looking at the males. Males do forage, but they forage really only for themselves. They don't really forage for the colony. I thought that the males were going to be worse at learning than the females. This actually wasn't the case, at least in this experiment. They were a lot slower, which I thought was kind of interesting, but despite being slower, performed just as well. I would love to delve more into males in the future. I kind of think that there are going to be differences there. They're just more subtle than what I was measuring in that experiment. But a couple of years ago now, I got interested in looking at the queens, because there's, like I said, this is such a critical stage of the colony life cycle, but one that has been so neglected when it comes to cognition. And I guess just to say that, we know that bumblebee foragers have got these amazing cognitive abilities, but at this stage, they can still rely on other individuals. If a bee makes a mistake, if she comes back to the colony and she hasn't managed to find any food, there's food stored in the colony, she's not going to die. Whereas at this stage, there's so much pressure on the queen to be as good as she can be, basically. Okay, and so I did this field work at 2 different sites. one site, which is basically a wet meadow that becomes a dry meadow across the season, and then another site in the desert. I also did this work during lockdown of 2020. And so it was done either in the field or sometimes in this basement where I was quarantining from my friends who had COVID. I mentioned this really just to say that there is definitely a time and a place for old school animal behavior methods. They turned out to be very useful during the pandemic. This is a Bombus vosnesensky worker. Again, very similar to what I showed you before. Here she is going to the wrong one and then finding the reward on that yellow strip. With all these bees, I first tested for their sucrose responsiveness. At least with honeybees, we know responsiveness correlates with learning ability and is kind of a way of measuring for motivation. I'm not going to talk about these results. I did not find that responsiveness related to learning performance. I'm happy to talk about that more if anybody's interested. But I then measured, I measured learning by differential conditioning. They had 2 forced trials where I gave them the S+ followed by the S-, followed by 5 choice trials where the bee is making the choice. And then I had 6 probe or test trials where neither stimuli were rewarding. And I found that the queens were better at learning associations than the workers. And so this is a group level learning performance. This is the proportion of the bees making the correct choice across the 5 learning trials, and the queens are performing better than the workers. And in that unrewarding test trial, the queens are also performing better than the workers. So I kind of set this up in talking about differences in foraging, and of course we're testing them in a foraging context, but there are many other things that differ between queens and workers in terms of the social requirements that they have. And so another group of bees that I also looked at were parasitic bees. So on the left here, we have vosnesensky, on the right, we have Van Dykii, and in the middle, we have Bombus insularis, which is a cuckoo bumblebee. And so these bees, it's not a perfect control, but it is a group that are foraging at this same stage, but without the same colony life cycle. So they don't produce workers, they only produce queens. They parasitize on the bumblebees colony. So the queens will go into another colony, they'll kill the queen, they'll usurp the colony, oviposit their eggs, and then the workers will rear their eggs to be new queens. But they still forage, just like queens do at this stage. Okay, and so this parasitic bee, the queens performed just the same, basically identical to the non-parasitic species. So I do think it's to do with these differences are due to the foraging requirements on individuals versus bees functioning as a colony rather than anything else. But I would love to look more at other cognitive abilities in these cuckoo bees. Okay. So another thing, of course, doing work in the field is that there are many variables that vary between these 2 different stages, right? That this is in the spring, this is looking at the queens, whereas the workers are out in the summer. This is a diagram of when the data were collected. So the desert sites before the meadow sites based on the flowering phenology, but also the queens are all tested before the workers. And so a lot of things can vary between those 2 stages. And so then I also decided to replicate this in the lab to just remove that component of experience. And so in the lab, I took our lab Bombus impatiens, and at the same stage of the colony, so towards the end of the colony, I took naive gynns and workers and compared them in their learning ability. This time I had to make the task a little bit harder because the original task I was using in the field, they learned way too quickly. They all got to 100% almost immediately, which I think is because they were naive, that the stimuli were just so salient to them. But so to make it harder, I used colors that were more difficult to discriminate. I tested one fewer training trial. And these bees showed the same thing. So the gynes, this is the learning performance, the gynes are performing much better than the workers. And in the test trial, only one gyne made a mistake once. They were basically perfect, whereas the workers are performing more around 80%. And you can see this is not a massive sample size for this kind of data, and it's still a pretty strong effect. Okay, and so now this year, going forward, I just want to understand more about the differences between the queens and the workers and their cognition. So my grad student Felicity is going to be doing an experiment this summer looking specifically at reversal learning. So one thing about being a bumblebee is that, you know, they live in these environments where flowers that are available to them can just change so rapidly, especially in these desert sites where you'll have these sudden blooms for maybe 3 days, and then the flowers disappear and there's other flowers that are blooming in a different location. And so you not only have to learn which flowers are rewarding, but also rapidly forget that information, not waste their time going back to those same patches, and then switch to foraging in a new lot of flowers. And so we're going to be, looking at reversal learning between the queens and the workers. I'm also interested in other modalities. So I've obviously focused on this visual modality, but I'm also interested in whether we see differences in olfactory learning. And so these are things that we're hoping to look at this summer. My grad student, Leah, right now is also looking at queen and worker brains. And so we got interested in how queen and worker brains might differ to each other, but also how queen brains might change across their lifetime, because the queens go through such a dramatic shift from being this foraging individual that has to learn all this information, this visual information, to basically living in the dark, you know, pumping out eggs. The requirements shift so dramatically. We're also interested to see if their brains change, essentially. We're going to be measuring learning in these 3 different stages. Leah's been doing this. And then we're also hoping to pair the learning data to the brain data. And so to do this, we're just looking at whole brain morphology, specifically focusing on mushroom bodies and regions that have been found to vary across different social roles in other insects. And so I guess the precedent for this is people have looked in honeybees and in osmia and paper wasps, some other species, and have found that brain morphology varies across different roles. But 1, those insects have very different life cycles to the bumblebees. And 2, the thing that we're doing here is really trying to pair the differences in cognition to potential differences in the brain. And we're doing this work. I am not a brain person. We're collaborating. We've assembled a dream team. So here on the right is Wolfie Gronenberg, who is the bumblebee brain guy. And Chris Jernigan from Cornell arrived in Austin yesterday. And so he's working with us today. We have our bumblebee brains. We're going to be staining them on the confocal, visualizing them on the confocal today. I'm really excited about this. So this is ongoing work. Watch the space. Okay, and then finally, just in the last 5 minutes, I wanted to talk a little bit about another theme of research in my lab, looking at anthropogenic effects on bees. So, as we all know, neonics are just about everywhere. We use them on, so many different types of food. And in a study that my postdoc Harry did in the last couple of years, we specifically looked at so much work is done using honeybees. They're kind of the model species for understanding pesticide effects on bees. But all the other bees are, of course, being exposed to pesticides. And so Harry did this meta-analysis where he showed that neonics impair reproductive output, colony growth, and foraging in bees that aren't honeybees. So we know that, we know from this and from other work that there are these negative sublethal effects on bees. And one that's been described a lot in bumblebees is effects on colony growth. And it's been suggested that some of these effects on colony growth are driven by effects on bees' ability to forage efficiently. They're not able to collect as much nectar and pollen. Of course, the colony is not going to grow as well. And it's been suggested that these effects might be driven by effects on learning. If a bee can't learn about which flowers are rewarding and how to get back to the colony and all the other things that bumblebees learn, then they're not going to forage as efficiently. In 2019, I summarized as part of another paper how many studies used different protocols to look at pesticide effects on learning. And most of them use olfactory PER, so where the bee is harnessed and they're learning an association between a scent and the sucrose reward. I think that PER is so useful for answering so many questions in learning, but when it comes to looking at pesticide effects on learning, I think it's not always the best protocol because we know that pesticides have so many effects on bees' motivation and ability to consume sucrose. And so when a bee is not sticking its tongue out in response to a scent, it could be that it hasn't learned that association, but it could also just be that she's not motivated to be drinking right now. And those motivational effects can be teased apart, but it's often difficult in such a kind of simplified protocol. And so I did an experiment. I wanted to know whether we could still see these effects when bees were free flying. And so here's the array we used. One color was rewarding for nectar, the other was unrewarding. And I found that bees that were exposed to a neonic there were all these effects on their behavior. They were less likely to forage, they were less likely to gain rewards once they were on flowers, they made less efficient visits, and they collected less nectar. But I found absolutely zero effects on learning. And so this is in the test performance phase. This is the control group. This is a 50% chance performance line. So the control group are learning, they're performing above chance. And then this is an increasing dose of imidacloprid, which is one of the most commonly used neonicotinoid pesticides. And at this dose, at the 50 micrograms per kilogram, basically half the bees were unable to fly. They were so impaired, but the ones that were kind of sadly dragging themselves to flowers were still totally fine when it came to learning. And so this was very surprising to me. I was not expecting to find this result. I repeated it with another experiment in the same paper. And then right around the same time, another study came out using Bombus terrestris and I was looking at acute exposure, they looked at chronic exposure, and they basically found the same thing, that foraging motivation was impaired, but not visual learning. And so these results together got me thinking, okay, this is a real thing. I don't think this is just chance that we're not seeing effects here. I really think there's something going on. And I wanted to follow up on this further. And so in thinking about why we didn't find impairment to learning when previous work had, one of the big differences, of course, was that a lot of previous work used harnessed bees, whereas we were using free-flying bees. But another big difference was the modality, that scent versus a visual cue. And so I then did another experiment where I had flowers that both had a visual and an olfactory cue. So for example, purple flowers with linalool, or blue flowers with geraniol. So the bee is now trained where there's two conditioned stimuli that they're using to determine whether there's nectar there or not. So yeah, counterbalanced, of course, half the bees are control, half the bees are treated with an acute dose of this neonic. And then in the test phase, I gave them both the S+ and the S-, but also stimuli where I'd put the color and the scent into conflict with each other. And I did this to look specifically at the type of errors that bees made. And so now on this y-axis, instead of looking at the number of correct choices, we're looking at the number of errors bees make. And so when they're going to one of the stimuli where both the color and the scent was the wrong one, we see very few errors. And that's what we'd expect, right? I just told you about how learning doesn't seem to be impaired. if we look at the number of color errors, so going to the flower where the color was wrong, they're making some color errors, but there's no difference between the control or the pesticide treated group. But now if we look at the scent errors, they're making fewer scent errors overall than color errors, which is not necessarily surprising. Bees generally have a harder time learning about scent than they do, have a harder time learning about color than they do scent. Scent is so salient to them. And clearly they're not making that many errors, so they're still able to detect scent in some way. But the key thing here is that the neonic treated bees are making more of these scent errors than the control bees. And so in terms of going back and thinking, okay, what could be driving some of these effects on colony growth? Is it learning, as has been proposed in so many papers? I really don't think it is learning. I think it could be olfactory learning, but I actually think that really this could be upstream of learning, and it might be olfactory perception or processing that is more messed up. So there was this nice paper that looked at the neural basis of neonic effects in the honeybee, and basically found that, yeah, this impairment is happening upstream of the brain. It's happening in the more peripheral kind of sensory olfactory parts of the antennae. Okay, and the implications of this are broad, right? So bumblebees, of course, are not the only bees that are using scent. All bees pretty much use scent, and not just when foraging, they're using it in communication. And bumblebees, they use it in the nest. And so I think that there are many implications beyond just foraging if scent perception and processing are impaired by neonics. And one thing that came out of the meta-analysis that Harry, my postdoc, did was that when we were looking at effects of neonics on bees that weren't honeybees, I'm sure this won't be surprising to you, but we really didn't find many papers on a broad variety of bees. Second to honeybees, bumblebees are the favorite species. So a lot of work on bumblebees, work on osmia, but really when we think about all the bees that are out there, we really know very little about native and solitary bees. And so now going forward, given what we know from honeybee and bumblebee systems, one thing that we're doing in my lab is looking at other bees. And so my grad student, Leah, is currently working with squash bees that she collects right here in Texas, and looking at the effects of neonics, but also on other insecticides that get used on the squash flowers that they visit, and seeing how this is impairing both sucrose sensitivity and feeding motivation, but also specifically scent perception. And squash bees they're very specialized on squash flowers, so they're not like bumblebees in that they're not visiting many different types of flower. They're really cueing in on a particular scent coming from the pollen. So this is the setup she has. We have a little kind of tea maze connected to an air pump where we either put in squash pollen or particular components of the squash pollen that are known to be attractive to squash bees. And she puts her little squash bee in here, and we can see whether they navigate towards the scent or how this is affected by different pesticides. And so Leah is just gearing up for she started piloting this last year, and now she's gearing up for the field season right now. Okay, so with that, I'm gonna conclude. I would just like to thank my absolutely amazing lab, including all the undergrads who have been wonderful this semester, my collaborators, my funding, and if you would like to see more about any of my research, please go and check out the website. All right, thank you. And now I will take questions.
Okay, thank you very much for this very interesting talk. Yes, are there any questions? So I would like to again remind you that you can raise your hands and then ask Felicity directly. So we have a question by Andrew Stropp.
Thank you so much for that really fascinating talk. I was really interested in the results which you showed, which I had not seen when you published them, with the scent and the visual learning that you just described at the end, and the differential sensitivity to the neonics. So would your assumption then be that there would be, I mean, I think the basic idea is that mushroom bodies are the seat of associative learning in insects, including bees. And so then would your hypothesis be that there would be like different neurons that would then be learning these associations, and that those neurons would then have also, you know, different like nicotinic acetylcholine receptor expression? Or basically, how would you, I mean, this does seem to a little bit contradict the idea that there would be like just a general associative learning mushroom body Kenyan cell type that could subserve any kind of stimulus.
Well, so like I said, I was looking at learning in my experiment, but really I think that the effects are happening upstream of learning. So I think that the information, the olfactory information, is already getting to the mushroom bodies and you know, in a scrambled format, basically.
I see. Okay. So, but I guess it could. I mean, the visual system has many more synapses actually prior to the mushroom bodies than the olfactory system, and there are plenty of nicotinic acetylcholine receptors in the optic lobes. So, I actually would imagine that the visual, but maybe that's also, well, no, because yeah. So I would actually, it's still surprising to me then with that explanation.
I also think that like, so every neonicotinoid activates the acetylcholine receptor slightly differently, right? There are all these different subunits on the different acetylcholine receptors. I was just looking at imidacloprid. It could vary across different neonics. And obviously acetylcholine receptors are kind of everywhere in the insect brain. And so I think that, I don't know, I definitely wouldn't want to speak to like one particular pathway, because I think that there's a
okay. So, but so there's no like obvious explanation that you, I mean, you propose it's the olfactory system differences, but I guess I would say, I would even imagine that the visual system would have more sensitivity to neonics than the olfactory system.
Yeah, I mean, like I said, that isn't what I found, and it's not what has generally been found in the literature. There is other stuff that has been found to be impaired. So, for example, spatial cognition, which does involve, you know, a lot of visual components. I think because so many, getting at the nitty-gritty is kind of interesting to me, but I think that because there are so many different components of behavior that are messed up by neonic exposure, it's kind of it's difficult to tease apart exactly what is going on in many cases. Ultimately, I also kind of think that the thing that matters is whether the bees can forage and like reproduce and grow. And those are the things at the end of the day that actually matter in terms of neonic exposure. In this case, I do think, and in, you know, taking the literature more broadly, I do think that olfactory stuff seems to be messed up more than visual stuff. But yeah.
Okay. Thank you very much. Yeah.
Thank you. I see we also have a couple of questions in the chat, but maybe we can first have Adam's question and then maybe alternate between chat and hand. So please, Adam, go ahead.
Okay, thank you. Thanks for the talk. I really enjoyed it. Well, I guess I have all kinds of questions because there's so much interesting stuff. But the one I wanted to ask, your work looking at queen versus worker learning. I mean, it seems, I've sort of always assumed this, but I'm not sure it would be true. But they invest so much more in the queens to make it through diapause, right? They're bigger bodied, they have all the fat stores. It seems like you would, or I would expect that they would also have essentially higher quality brains, whatever that means. That it's, you know, the workers are somewhat disposable. They're probably better off making maybe faster decisions and accurate decisions in some circumstances. Whereas, so I guess you're doing this work, and I'm interested to see what the confocal stuff says, but it seems like from a colony level perspective, you're a lot better off, you know, investing a lot of extra energy in queen neurons because the cost is relatively low. It's just one individual. Whereas the reverse would be true for the workers, right? If you invest a lot in worker neurons, let them do an 80% job, but have a lot more of them than, investing that extra bit for them to do well. Is that what you're seeing, or is that the kind of stuff you're not going to find out until you get the confocal results back?
Oh yeah. Today, after I finish giving this talk, I'm going to lab, and it's day 1 on the confocal. So yeah,
so you really literally are just okay. Okay, cool. No, that's really interesting. Then I'm sorry. I guess that was more an observation than a question. Then I'm sorry. I'll wait.
Oh no. One thing that you mentioned though was, and I don't know if this is something you were thinking about when you mentioned it, but the kind of speed and accuracy.
Yeah.
Which is something that we are hoping to look at as well. Anecdotally, the queens were crazy fast and very accurate, which is kind of interesting because within workers, you see a speed accuracy trade-off.
But not in the queens.
Well, I don't know. This is me saying based on my kind of observations from doing this, but I would like to measure it as well.
Oh, cool. That's really interesting. Thanks.
Okay, then I'm reading now questions from the chat box, but let me just ask a question regarding the queens and the workers. Did you test nectar and pollen sensitivity for the queens? And do you expect that that might be also different from workers?
So I specifically collected queens that didn't have pollen loads because I didn't want to be messing up any early colony founding in queens. But no, I didn't measure. I mean, I measured sucrose responsiveness in both queens and workers, but I didn't measure any kind of pollen. I don't even know how you would actually do that. Who asked that question?
Yeah, no, I asked it. What?
Oh, you're asking it. Sorry. Okay.
No,
Yeah,
no,
how would you measure pollen sensitivity?
No. Okay, let me kind of. I mean, okay, you could do these learning experiments that you did. Maybe you cannot do this with the queens. So right. So my point was, so to say, whether nectar and pollen sensitivity is depending on the physiology of the animal. So in honeybees, foragers are basically not eating pollen at all. So they kind of like a carbohydrate kind of physiology. So kind of like, I don't know whether Andres kind of talked about, when we talked about pollen sensitivity in honeybees, whether nurse bees, for example, needing kind of to feed kind of pollen, kind of to have food for the brood, might show a higher sensitivity to pollen. Now comes the queen, kind of like the queen probably needs protein to produce the eggs. So point what I want to make is kind of like how to say the strength of the reward, nectar versus pollen, might be related to the physiology, which kind of probably people haven't kind of thought about that much, right? Because they were kind of starting to do these learning experiments with honeybee foragers.
Yeah, and so I think in terms of, okay, so I guess a couple things. With the pollen foragers, with the workers, we did note whether they were foraging for nectar or for pollen and whether they had pollen loads or not, and that didn't seem to affect their learning performance. I'm just going to share my screen again quickly just to show this one graph, because I think it's kind of interesting and maybe speaks to your question a little bit. So this is the sucrose sensitivity data from, these are 2 different sites, the meadow and the desert site. And so at this one site, this is, how likely bees are to respond to a sucrose concentration of increasing concentrations. At this site, there's no difference between the queens and the workers. Here, the queens were a bit more sensitive. But you can also see up here, the insularis, the cuckoo bumblebees, are just crazy responsive. They're responding to everything. It doesn't seem to translate into their learning performance. And more generally, if we correlate like sucrose sensitivity and learning performance, there wasn't a relationship. My grad student Smriti is actually collecting data with workers right now for nectar foraging versus pollen foraging workers to see if we see these differences that are so well documented in honeybees in terms of the differences in sucrose sensitivity, and like foraging role. And in the field at least, and she's now replicating in the lab, we don't see the same effect as has been found with honeybees. And so I do think it's whatever's going on with bumblebees is different to honeybees.
Okay, just one comment. I think in Drosophila, it's already kind of known that insulin pathway has an effect on learning and memory. And insulin would be kind of like one output pathway of the nutritional status, right? So, okay, good. But now I'm kind of going to the question in the chat. Let me start with Gard Otis. Can you better explain how you presented the pollen to the bees in your test? I see a rod on an artificial flower. How is the pollen added to the rod?
Okay, so that is just, yeah, like a pipe cleaner bought from Michaels. I think Michaels think I'm insane because I bought like 1,000 brown pipe cleaners from them to make the most boring crafting project ever. And we came up with a system where we had perspex that we drilled like a very specific depth into. And so we had these little divots. And then we kind of pushed the pollen into these divots so that each divot held a specific quantity of pollen. I'm now forgetting exactly how many milligrams it was. And then we basically just dipped the pipe cleaner into the divot. And it was like the most efficient way to get kind of the same amount of pollen on each anther, basically.
Okay, and then there's a question by Anthony Antonucci. Do bumblebees practice floral fidelity when foraging similarly to the honeybees, or will they visit many different types of flowers on one trip? Basically, flower constancy in bumblebees.
Yeah, so they show constancy like honeybees. You know, they will still visit many different types of flower on one trip, but they also generally, yeah, show constancy.
Let me just add one additional question. Anthony has kind of like 6 questions or so. So he's really busy. So do the foragers communicate to other hive foragers as to the location of nectar and pollen, like the honeybees? And how large is the foraging range of a bumblebee? I mean, differences between bumblebees.
Yeah, okay, no, they don't have the waggle dance. That is a crazy, weird honeybee thing. And the foraging range, it can be really big. So bumblebees will forage basically as far as they need to. If there's a flower patch right there, they'll go to the closest flower patch. If they have to fly a few miles, they will go a few miles. Also, I noticed that Margarita has her hand up as well. I don't know if you want to. Cool, go for it, Margarita.
Yeah, hi. So I have a couple questions. One question is actually related to Anthony's question, right? Yeah, they don't have waggle dance, but they do have the footprints. They do have like food marking with the chemicals. Does learning scent, because learning scent can also be applied to learning the food marking scent. Do you think that the presence versus absence of food marking affect the results of scent learning? Did you control in any way for example, like you released 17 bees in a row on the same patch and some flowers were previously visited by another bee? Is there a possibility that the food marking by that bee might affect subsequent results?
So yeah, bees definitely do use scent a ton. And so that's something that we're always thinking about in all our experiments. So, you know, bees are always tested individually so that there's no use, they can't use like social scent cues. And then between every trial, we take out the flowers, we replace them with clean ones, and we clean them with alcohol.
Okay, so there is no possibility that one bee that was tested, she landed on something and then kind of left a cue for the next bee to land on the same thing?
No.
And another question is concerning the differences between queens and workers, and probably the differences among workers, because there are some older studies that show that bigger bees have greater antennal sensitivity. So queens are obviously big, like they are really big bees. The question is if that you think can come into play when you see that queens are much better at learning, and whether, for example, if you look at workers, whether, for example, bigger workers are better at learning than smaller workers.
Yeah, so I think that's really interesting. And, there's one paper by Wolfe, I think Nigel Rain also had a paper, where they found that larger bees learned better. I look at this in every single data set I collect, because we always collect size as well, intertegular distance. And I actually have never found this relationship once you control for the fact that larger bees can make more visits on a given trial because they're larger. Because so often, they're controlling how many flowers they visit within a foraging bout. I'm pretty sure, but I'm not saying that that's not the case. I'm just, so I don't know, basically. Like I haven't found that with my bees. And in terms of just the size, I think this is something that, we're kind of interested in when it comes to the brain stuff. Is it that they just have larger brains, but everything is proportional to the worker brains? Or does the proportional investment in different regions differ?
Thank you.
Thank you. Yeah, I think Johannes Schwete who was here before, they have a study where they looked at the number of antennal sensilla depending on the size of the bee, and they find that the larger you are, the more sensilla you have on your antenna, and the more sensitive you are to odors. So I had the same question actually as Margarita, if that then maybe would lead to a difference in learning ability that is somehow related to size. But it's interesting that you don't find that.
Yeah, and I mean, like I said, it's not something that we've ever explicitly tested for. It's always been like, oh, we should control for this as a covariate in our, and so, I don't want to come out too strongly in that statement, but I think that's interesting about the sensilla and something that I've written down as a thing to look to follow up on.
Okay, let me see. There's okay, Anthony has so many questions. Let me kind of select some. So here's one. Are cuckoo bees only interested in nectar when they forage, since they utilize a bumblebee nest provisioned hive to lay the eggs?
That's a good question. So not a lot is known about cuckoo bees in general, but I'm going to hazard a guess that that's when they're foraging, is when they're trying to find a colony. And then once they find the colony, they're probably just eating the provisions that are in the colony rather than going out to forage again.
Okay, then he has another question. I mean, I'm not reading all, but so can viruses be having an effect on bumblebees' ability to learning and foraging behavior?
Whew, this is like a qualifying exam. I think there has been work looking at infection in general, both viruses and parasites. I think generally speaking, we do find impairments to learning. I think often it can be difficult to tease apart effects on motivation, like I mentioned, that like if a bee is feeling sick, it's going to be less motivated to feed. And so sometimes what people are detecting is a motivation, which is then affecting learning ability rather than probably a direct effect on cognition.
Okay. Is there any hand raising? Otherwise, I'm reading another question of Andrew.
There is a question by Natasha.
Hi. Thank you very much for your very nice presentation. I learned a lot. You mentioned at some point that you tested the learning abilities of male bumblebees. And I don't know if you're aware of this paper from Stephen Wolfe in 2009, I think, which found similar results. It was pretty consistent with what you found.
Yes, I do. There were, I want to say maybe 4 papers on male learning before the study that I did. Most of them didn't find differences. Some of them found differences in some contexts. The thing that we were doing that hadn't been done before was like collecting wild males and females. So ones we know had kind of gone through all the normal things that they would go through in their environment. So yeah, so I guess I feel like there maybe aren't big differences, but none of the studies I read nor my study really got into kind of super nitty-gritty specifics in terms of really fine scale discrimination or more complex, anything beyond just kind of pure discrimination learning. So I do think that it would be cool, for example, to look at potential, say, speed accuracy trade-offs, that kind of thing, because my males, they just kind of you just see these differences. They kind of saunter along like in a very unmotivated way, like, oh yeah, I guess I'll just choose this one. Whereas the females are so driven. They're like, okay, I'm headed towards the blue right now. And so I do think that there probably are some differences there, just ones that, yeah, haven't been detected yet.
Yeah, I also think it would be very interesting to look at the speed accuracy trade-off in males. And I'm working with male at the moment, but on a different topic. And I also found that they don't really care about the artificial flowers like the workers do. But it makes sense if the males are only foraging for themselves. And my other question was, because I think you mentioned that regarding the neonicotinoids, in honeybees, it has only been done on learning and no one has done experiments on responsiveness, for instance?
On sucrose responsiveness? No, they have. There has been a fair amount of work, often in like a PER type setup, looking at sucrose responsiveness. And that is something that shows up again and again, that bees that have ingested neonics are much less responsive to sucrose.
So it goes with your hypothesis of perception, doesn't it?
I think that there are many things that are going on. I think that neonics make bees less responsive to sucrose, but I also think it still impairs something to do with olfactory perception and processing.
Okay, thank you very much.
Okay, I have a question, just a technical question. When you tested for the 7-day memory retention, how did you do the experiment? I mean, did you not provide any kind of feeder in the arena so that they had to eat in the hive, or did you kind of presented different kind of feeders in the arena that were not rewarded? Or
that's a good question. So no, they didn't have experience between learning and then being tested 7 days later. I forget now if they had like a white-wicked feeder that they were going to, or whether they were just in the colony box the whole time. But if they did have experience, it was very limited experience. And so that's a good point, that it could be that if you're having like many more learning events in that time, you forget that information because it's no longer relevant. I think it would be cool to do something, now that you're saying it would be cool to do something like having many learning events when they're foraging for nectar, but they haven't collected pollen for 7 days. And now when they go out to collect pollen, do they still have that memory, or has it been, yeah. I think there's so much, because so little has been done with pollen in general. I still think that there's just so many questions.
the other way around. I think actually learning and memory in honeybees and bumblebees is so dominated by the PER conditioning paradigm, and people haven't kind of thought about ecological kind of factors that might kind of get into learning and memory, right? So kind of like similar what you said, we were kind of discussing in my lab, kind of what would rain do to learning and memory, right? So we let it rain for 7 days. Is the memory still there or not, right? And kind of like compared to a feeder situation where the feeder is not providing any reward anymore, right? So in the second one, you would kind of like expect a decline in learning, whereas when it's rain, you would kind of like the memories should be stable.
Mm-hmm, mm-hmm.
And the same as when you are talking about nectar and pollen, which is kind of like actually nutritional kind of physiology that plays a role in learning and memory. So.
Mm-hmm, mm-hmm.
Okay, Margarita has another question.
Wait a second. Yes. Hi again. Thank you for the great talk and for answering all the questions. I have another question and a comment on males. So like even from, my own personal observations in the field, they spend much more time on each flower, like workers that like each flower takes a few seconds. Males spend a really long time on each target. Do you think it's a motivational effect that they don't hurry anywhere, they don't need anyone to provide for, they just can sit there and forage for themselves and not care about anything else?
Yeah, and I think also males are often hiding out on flowers waiting for gynes to land.
Yeah, that makes a lot of sense. And now the question that I have from like dissecting male heads and female heads, it seems that males have a much smaller brain, but apparently comparable learning capacities. And this raises the question, how much brain do you need for that at all? Like, how much does the brain size matter?
And that's even, because I know that males have got larger eyes, which often makes their brain like smaller, like relative to the whole size, but is the absolute size of male brains smaller than female brains?
I mean, it seems that they don't have not only larger eyes, they also have huge labial glands. And it seems like that is what is taking up the most of the head volume and not the brain. Like on honeybees, there is research that actually there actually was volumetric research on male brains, and they were shown to be significantly smaller than both worker and queen brains. But honeybees are kind of abnormal, because in honeybees males are completely like useless. They don't know how to eat. They don't know how to. They pretty much don't know anything than how to find the queen and mate. But in bumblebees, it's apparently not the case, but the brains are still somewhat smaller. And the question then rises, how much does brain size matter?
Wait a minute. Finding a queen can be a difficult task. So don't make it kind of like too simple that finding a queen is just done like that.
Yeah, it's a good question. You know, I haven't actually been thinking about the males much for a while, but now you're making me wonder if we should just be taking some male brains as well.
Okay, so there's another question by Anthony. In your research, were you able to differentiate how first-time foragers from the hive do and know when they go out and forage as opposed to an experienced forager? Okay, that's a question I don't really understand. Do you? Maybe let's kind of like, Anthony is still there. Maybe he kind of like is brave enough to kind of ask this question. Anthony? Okay.
I think I understand this question. I think basically cool. we tag bees and we track what they're doing. Depending on the experiment, sometimes we define experience based on number of visits or amount of time. But yeah, we do track individuals and see how much experience they have.
I have a question that maybe you've answered. We have seen in Andres' talk that in honeybees you have this division of labor where some workers focus on pollen and the others focus on nectar. I was curious about how that is in your bumblebee species. Is there any division of labor in the same sense? And if not, why not?
Yeah, I think this is super interesting because honeybees have such stark division of labor that bumblebees don't have, but they do specialize. They specialize in different roles in terms of nurse versus forager, but they can switch. So if you take away some foragers, the nurse bees will step up and become foragers pretty quickly. And then within nectar and pollen foraging, bees will specialize, but not all the bees, basically. Like when I'm trying to keep the colony in a state where they're collecting both nectar and pollen, so I'm keeping them kind of low on both nectar and pollen, maybe around half the bees will collect both. 25% will just collect nectar and 25% will just collect pollen. And so basically to say that there's definitely more kind of flexibility to switch between the two than honeybees have, but there is still some often short-term specialization.
Yeah, I was wondering if maybe that is related to the smaller colony size of bumblebees, that
Oh
because
yeah,
they have
no, I
very
bet.
small colonies, at least from a kind of an organizational point of view, it would be maybe risky to have too strict specialization if you only have a few dozen foragers, whereas if you have thousands, then maybe you can have a very strict division of labor.
Yeah, and I think this is really interesting, right? In terms of thinking about the differences between honeybees and bumblebees, and yeah, the giant colonies that honeybees have where they can specialize so much versus bumblebees where every individual has to be a little bit more of a jack of all trades. I think there's lots of interesting questions to be asked there about how cognition varies in those two different setups.
Okay, last chance for questions.
Another one from Anthony.
Mm-hmm.
Does outside weather conditions have any effect on the foraging learning and on the foraging, learning, and behavior? They endure more harsh weather conditions than honeybees. Okay, let's in the mountains or in the northern.
I'll just like finish on like a fun anecdote that is, I feel like bumblebees like represent the humans who live in the locations where they are, because in the UK it rains all the time and people will just go out and do anything in the rain, and you see the bumblebees just like foraging in the rain. Whereas when you are in California, people will be like, oh well, it's not a perfect sunny day, I guess I'm not going to go skiing today. And it's the same thing with the bumblebees, because the weather is so nice so much of the time. As soon as you have a little bit of rain, they're just not going out and foraging. I don't think it probably relates to their ability to learn, but their motivation to forage is definitely affected by the weather.
Okay, cool, yeah. Mm-hmm.
Okay, thank you very much for these interesting questions and the answers. Thank you again, Felicity, for your presentation. And I hope to see many of you next week.
Thank you.
Okay.